Well, welcome everybody. My name is Professor Alex Leff. I'm one of the Deputy Directors at UCL Queen Square Institute of Neurology, and it's my pleasure to be introducing our two relatively newly minted professors today. They've got quite a lot in common, as you'll see. Firstly, you'll have noticed that they're both from the same department of the Institute of Neurology, the Functional Imaging Laboratory, or PHIL as it's more affectionately known. And the PHIL has really been a powerhouse of producing highly talented neuroscientists over the years. It's unparalleled really in terms of imaging neuroscience. And I was lucky enough, in fact, to start there for the first 4 years of my first academic appointment. And I was asked to do a talk in Germany, and my interest is in language, and the person who invited me rather sort of hyperbole introduced me as one of the world experts in language, to which I replied I wasn't even the best known expert on my floor. Just to say a little bit about today, each lecture has a vote of thanks. So Martina, Callaghan, who's the current head of the PHIL, is going to give vote of thanks to our first Peter, and the second Peter is getting Professor Cathy Price, who's the previous head of the PHIL. You're all invited to stay for both, in fact encouraged to stay for both talks, but we are going to have a brief change of ends because there's some younger people coming in, I think, for the second talk. And then when we've finished, which should be at 7 o'clock pretty promptly, you'll all be invited for drinks and snacks in the foyer outside. So to our first Peter. Peter started off, he's going to tell his own story, but he started off with his first degree in Birmingham in artificial intelligence and in computer science, and then did the first of his two master's degrees there in natural computation, then went into industry for a little while, and then came to UCL for his second master's degree in neuroscience, and before joining Eleanor Maguire's group to do his PhD on the hippocampus, which I'm sure we'll hear more about. And he's really been a PHIL stalwart ever since then, pioneering a lot of methods that we'll hear about. Indeed, he's been chair of the methods group at the PHIL for over 3 years now, and has started his own neurovascular modelling group. He works very collaboratively, both within UCL and also outside UCL, particularly with his recent MRC Career Development Award, which he's going to tell us a lot about. I couldn't help but notice, I didn't know this about you, Peter, but you were involved in something called the Great Brain Experiment. So it didn't leave his software roots too far behind, and he developed an app a little while back to sort of encourage people to understand more about neuroscience, and collected data on over 100,000 participants. I think he tricked them into doing some tests. Anyway, without further ado, Peter's going to tell us all about how to detect neurovascular dysfunction. Welcome, Peter.
Thank you, Alex. Thank you, Alex, for that very nice introduction. Over the next 45 minutes, I'm going to set out a unifying mathematical account of the brain and how we measure it, starting with the voltages and conductances of neural masses, proceeding through connections and synapses, to the measurements we make using magnetoencephalography, through the blood flow and deoxyhemoglobin response, to the measurements we make using fMRI, and linking from individual subjects to groups, using necessary proofs and derivations where required. Only joking. Over the next 45 minutes, I want to do the opposite of how a slide like this might make you feel. I want to get across how mathematics and mathematical modelling can simplify things, how it can enable us to cut through the almost unimaginable complexity of the brain, and tell really simple stories about how the brain might function and why it goes wrong in neurodegeneration. Sorry, let me put something more calming on the screen while I'm telling you about it. The key thing about having these mathematical models is they enable us to assign a number, a statistic, to say how good that explanation is. And it allows us to trade off different theories, different hypotheses, and work out the best understanding for why the brain goes wrong. And I'm going to be focusing on Alzheimer's disease in this talk. And those kind of formal methods allow us to disentangle different ideas in a nice principled and clean way without requiring arguments, fighting, or acerbic letter writing. So there is a tradition in these talks that we start with a little bit of autobiography. So I'll tell you a little bit about how I got here. Thank you in advance for your patience on that. And then I'll talk about the methods group and the neurovascular modelling group, the two sides of my research life. And having introduced the aims of the neurovascular modelling group, I'll talk about the methods we're using to investigate the brain and where we're going in the future. So I knew from a very young age, actually, that I wanted to become a magician with a side interest in ventriloquism. And the many, many hours that my parents spent watching my magic shows have not been in vain, because a lot of those same skills I use every day now as a scientist standing up in front of audiences like this. But as hobbies go for children, that hobby only lasted so long, and something else came along that was even more exciting. There were these little boxes of lights called computers. This in particular is what a computer used to look like in the UK, a Sinclair Spectrum. My parents had this, but they didn't have the tape drive that went with it. So what it meant was you had to program the computer using the book, the programming book, and then when you unplugged it, the program you'd written was lost. And as a very young child, I thought that was the point of computers, and for some reason I enjoyed that experience. So I spent a lot of my time, much earlier probably than I should have been, learning how to program computers. Americans had Apple computers, you might have heard of them. British, we had the Apricot. Had a calculator built into the keyboard. Amazing machine. So I did a lot of computing. I got really into artificial intelligence, computer science, robotics as a child. My parents never said, step away from the computer, you should probably go and learn to play football. And that early parenting decision, which may be considered questionable, is the basis of my entire future career. So I will say many things I am grateful for, and that is one of them. So it was perhaps unsurprising then, I went off to university and I read artificial intelligence and computer science at Birmingham. And for the first time in my life, I was out-nerded. There was other people there who'd written their own operating systems in assembly code and this kind of thing. It was very exciting for me as a computer scientist. And I learned about these things called artificial neural networks. It was kind of a strange time to be doing AI, because AI had been really massive in the 90s. That's when the core algorithms like backpropagation were invented. By the time I got to uni in the early noughties, it kind of slipped into, it had stagnated as a field. If I told anyone I was doing artificial intelligence, like at a party, I get a kind of confused look and I get the question, is that something to do with aliens? So I made, I think rational for the time, the very sensible decision that AI probably doesn't have a future and it's not going anywhere. That is a decision that has aged badly. But it did mean I got more interested, less interested in the artificial neural networks and more interested in the real ones. And I remember a really formative moment where the BBC Reith Lectures on Radio 4 in 2003, presented by Vilana Ramachandran, the very famous California University professor of neuroscience, very famous for the rubber hand illusion. And he has this remarkable delivery. And I remember just listening to him talking about these parts of the brain called the superior colliculus and the medulla oblongata. And I was sold. And I don't know if it was his delivery or the content, but I knew brains was the direction I wanted to go in. So I did think, though, that before I retrained and learned all about brains, I should first try having a real job. So I went and worked in the software engineering, software consulting industry for a company that is now defunct. Nothing to do with me. They were bought out and very successful. But I have no regrets about those couple of years I spent in industry. I learned a lot of very good professional skills in terms of software engineering and professionalism, working with clients. And for any of my colleagues, I would encourage having some time in industry, but definitely then come back and share what you've learned. And I was all set up to go and work on a new client, the ITER nuclear fusion project in the south of France. Their magnets are even bigger than ours, at 12 Queen Square, by the way. But there was a downturn in the economy. My project got cancelled, and it was my moment. I quit my very sensible job in IT and came to Queen Square for the first time. It was the first to undertake a master's in neuroscience. And it was the first time I stepped into this place called the Functional Imaging Laboratory, the PHIL, aka Neuro Disney. That was a term I heard several times over the years, and I now have an understanding of why it got that nickname. And it's not because of the amazing machines we have to scan people's brains, although that's definitely part of it. And it's not because of the amazing science, the fact that the whole field of neuroimaging analysis was born here, although that's part of it. And the amazing support staff, that means we can just do science without having to think about admin and bureaucracy, although that's probably part of it. But for me, it's very much about the people. So I've been there for 15 years now, seen a few generations of students and postdocs. But what is consistent is the kind of attitude and the culture and the way of being that people have. A real openness, a real warmth and excitement about science and a desire to talk about it. I've met some of my closest friends whilst being at the PHIL. It's a wonderful place. And all the work I'm going to talk about today is work that I've done here. My first entry into neuroscience was with Cathy, who supervised me as a master's student. And I learnt about how we can investigate the differences between people after they have a stroke in terms of the connectivity in their brain. And we did this project that I remain very proud of. I think it's a really exciting piece of work, using a technology called dynamic causal modeling to show how a patient was able to use a different pathway in her brain compared to healthy controls. That technology, it turns out, would be foundational for all the work I've done since. So I think the message there is, well, number one, thank you, Cathy. And number two, if you have the opportunity to take on a master's student, you can really change someone's life. So this was a very firm foundation. Just as that project was ending, a vacancy came up in the lab of Eleanor Maguire to study the hippocampus. And Eleanor was very much the best PhD supervisor I could have hoped for. She taught me how to be a scientist, and she taught me how to be a cognitive neuroscientist in particular, and gave me an understanding of psychology, which I didn't have at all from my background. I learnt an awful lot from her, and in particular, I learnt a love for the anatomy of the brain, and the hippocampus in particular, this deep structure in the brain that's responsible for many of the things we do on a daily basis. And this review paper, I know she was particularly proud of. The key kind of take-home message of it was that there's this common bit of the brain, this common bit of the hippocampus, that in study after study is revealed using functional MRI. That little bit of the brain lights up, regardless whether you're perceiving a scene like the one in front of you, or if you're imagining a scene in your mind's eye, or recalling a scene from your past. And kind of the interpretation, the take-home from this, is just as a scientist builds these mathematical models to understand the brain. So the brain is making models of the world around it that it can then use flexibly, whether you're looking or imagining or recalling. And it seems that this very precise bit of the hippocampus, called the anterior preparacebiculum, is a crucial part of that circuitry. Eleanor stayed a mentor for many years after my PhD, and we all miss her very deeply. That very, very good start was just one of many good bits of timing. Another very good piece of timing is just as I was finishing my PhD, a vacancy came up in the methods group at the PHIL, which would allow me to indulge my technological and computational interests and go much, much deeper into how those analyses, like the ones I just showed you, are generated. The methods group isn't like other research groups. It is very special indeed. It was founded by Carl Friston in the early 90s, and since then has developed almost all of the key technologies that are used in every neuroimaging study ever published. So anytime you see an image like this, where we have colored blobs on a brain, either showing brain function or changes in the volume of the brain due to a condition, that is using inventions and technologies from Carl and John Ashburner and their team at the PHIL over the years. And those methods, which together are called statistical parametric mapping, we make available in a piece of software that goes with it. And I've been very honored for the last few years to be chairing the methods group, developing this software and developing these methods. And It is a fantastic team who give up their time alongside their research to contribute to this software that's given away freely and used by researchers all around the world. This is a really exciting year because we're celebrating 30 years of continuous development of the software, and we've been doing some things to mark that occasion. And I'll just spend just a couple of minutes mentioning those, because I really want to also thank the members of the team who are here in the room and watching online. So for our 30th anniversary, the first thing is in the years leading up to this, we've really completely changed our methods of software development. We're using approaches that are open and transparent and enable more people from the community to contribute than ever before. And we've just released the first new version of the software which incorporates those practices. This is the hard work of a lot of people in the team, and it's something that we're very proud of. Alongside that, one for my neuroimaging colleagues here, SPM Python has generated a lot of excitement just in the last few months. So Python is the most popular programming language in the world. There's often been some consternation that our software is not written in Python, and my colleagues Johan, Yale, and Julia have done amazing work in making SPM available to Python users. And that's going to broaden the number of people who can use SPM, especially as it's all completely free of charge. We've modernized our courses, so one of the real highlights of the year is that people come from all around the world to learn about SPM in this very room, as you can see. And that modernization, the hard work of Olivia, who's with us today, has meant that the courses are much more entertaining and accessible and a lot more fun. We were recognized by the biggest conference in our field for our work with an Open Science Award at the Organisation of Human Brain Mapping, collected here by Guillaume Flandin, who was my first line manager and steered the SPM ship for many years. And we've just got back from a big jolly in Australia, in Brisbane, again, the biggest conference, OHBM, and we were able to have a big celebration of 30 years of the project, where we held a nice big event. And we didn't pay for this, but the conference expressed in their own words the love that they have for our project. So the bottom line I just wanted to highlight is that the project's going really well, and it's a really nice team effort, and it's made possible by the culture at the Phil. SPM isn't everything we do in the methods group. The other thing we do is we work with researchers all around the world on the analysis of data, trying to fill the gaps where mathematical modelling techniques don't exist, and we create those models where needed. So ever since Carl created SPM, there have been these weekly sessions where anybody can come from anywhere in the world and ask questions about their analysis and get feedback. And this has been something of an apprenticeship for me for the last couple of years. Carl's practice is that he asks me to have the first attempt at answering people's questions, and then he embellishes with what I should have said, what I should have seen, and that has been a gentle tutelage that has been absolutely invaluable and taught me a huge amount. This word cloud just shows the titles of papers that have been published as collaborations that have come out of those meetings. And the projects that I've really enjoyed the most over the years that stand out in my memory are those that had some clinical application. PTSD, schizophrenia, aphasia, Alzheimer's disease, Parkinson's disease. There's a real satisfaction in working on projects where we can gain insights into clinical disorders. And that really motivated me to start putting in for funding applications, trying to apply the modelling techniques we've been building to gain a better understanding into neurological and neuropsychiatric disease. And that led to then the starting of the neurovascular modelling group. It's going to be a slight gear change now onto a more serious topic than modelling. I first got interested in Alzheimer's disease when I was a PhD student in Eleanor Maguire's lab, because a big feature of the disease is that one of the key pathologies, tau pathology, moves through the circuitry of the hippocampus and out into cortex. As well as, we all know it is a devastating condition. There's about 982,000 people in the UK with dementia. It's expected to rise to 1.4 million by 2040. And of the different dementia diagnoses, Alzheimer's is the most common. An attempt was made about 10 years ago to make a physiologically grounded definition of Alzheimer's in terms of the markers that you could measure in the brain. And that gave rise to the ATN framework. 3 key features that defines if someone has the disease. The first is amyloid, a toxic peptide that builds up in the brain, either as soluble form or it clumps together into these plaques. The second marker is tau. Tau is the scaffolding of neurons, and in Alzheimer's disease, it becomes tangled and forms these toxic proteins that build up. And both of those 2 markers can be detected from the cerebrospinal fluid or from using PET imaging. And the third marker is neurodegeneration. And if you compare a hemisphere of brain from a healthy brain compared to one with severe late Alzheimer's disease, you can see this very clear difference. In particular, there's a shrinkage of the hippocampus and enlargement of the ventricles. So those 3 features were originally proposed, but in the years following, many additional letters were proposed to add to that ATN framework, other markers that are important for understanding the disease. And a key one among them, the one that I'm focusing on, is V. V is for vasculature. So it turns out that the blood vessels in the brain are very important for understanding the disease. Let me just give you a super quick primer on blood vessels in the brain, and then I'll come on to its relevance for understanding Alzheimer's disease. So we talk about something that we refer to as the neurovascular unit, which consists just boiling things down in a very, very reductive manner. It compartmentalizes the brain into 3 parts: neurons, the blood vessels, and the stuff that links them together. And that subsumes a lot of different mechanisms, direct and indirect mechanisms. It's really important that that coupling is working efficiently. So the blood vessels have to dilate to allow oxygenated blood to come in, and it has to constrict to stop that blood when it's no longer needed, and it has to happen at the right time and in the right positions in the brain. The way it's able to do that, if we take a cross-section of an arteriole or an artery, there is muscle within the actual blood vessel. You have pericytes, and you have these smooth muscle cells that enable the neurons to signal that they require additional blood or less blood. So when I was a PhD student with Eleanor, if you had told me that, I was studying imagination and memory and consciousness, if you told me that the blood vessels were interesting, I would have said, well, no, it's just plumbing. That's not very interesting. But actually they are fascinating machines. They are much more than just pipes. That coupling, as I mentioned, is very precise, and I like to call it a precisely orchestrated dance. It is beautiful to watch. I always show this video because I enjoy it so much. This is an ultrasound cross-section of a mouse brain. And what you're going to see is the area here of the mouse's brain that senses movement in its whiskers, what's called the barrel cortex. You're going to see the blood flowing. Blood flows from the surface down into the cortex in yellow and back up to the surface in blue. And then you're going to see what happens when they start tickling the whisker. And you'll see the change. So here's before the tickling. You see blood coming down into the brain. And now they've started tickling the whisker, and you can see that huge increase, both in the rate of flow of blood, but also the volume of the blood. There's more blood. And now they've stopped, and it goes back down again. Isn't that beautiful? I think that's and that's happening in your head constantly. So if you hear a bang, the auditory parts of your brain are demanding that blood. The visual parts, if you were to see something. That has to work well. In Alzheimer's disease, that process, that neurovascular coupling breaks down. The relevance is twofold. First is that it may well be a prelude to the ongoing development of the disease. This is a study that I like very much. I think it's very elegant, from Oxford University, looking at people who are at particularly high risk of getting Alzheimer's in future. They have APOE44 gene. They're only in their 20s and 40s. They don't have the ATN yet. They don't have amyloid, they don't have tau, they don't have neurodegeneration. But what they do have is a huge reduction in their what's called cerebrovascular reactivity. In other words, the movement of the blood vessels. So even at this very young age, before anything else has happened, there's something going wrong with the blood vessels. And that's really tantalizing, because it might mean at the very early stage, there is a chance to intervene before the other markers have come online and before symptoms have begun. So we don't know what role vasculature has in the early stages of Alzheimer's disease. We just know it has a role, And the question is, could that be made use of, either for diagnosis or for treatment? The other role it has is it's part of the disease process itself. So amyloid builds up in the tissue of the brain, but it also builds up in the blood vessels. So here's a blood vessel, and you can see in pink the muscle that I mentioned that allows it to contract. And these little blue dots are amyloid. And what happens over time is that the amyloid takes the place of the muscle. And what happens then is that the blood vessel can't move anymore. It becomes stiff and it becomes leaky. So there is a need to identify that this is happening and treat it. But a great challenge is that there is currently no way to diagnose this condition, cerebral amyloid angiopathy, in life. It can only be identified when it's really far down the line and it's caused bigger bleeds. And it can be diagnosed post-mortem. But there isn't an imaging approach to detect that in life. And just to say that what we're really interested in mainly at 12 Queen Square is cognition, is thinking. And these vascular problems cause cognitive problems. So this is a large-scale post-mortem study showing how if you just look, so what they did is they assessed people's cognitive function, and then when they eventually passed away, they just looked what pathology was in their brain. And in red are the pathologies that are vascular, and in blue are those that are not vascular. And you can see by far the most common pathologies are vascular, including this one I just mentioned, CAA. And then they did a factor analysis and showed that a great deal of the variability in people's cognition in older age can be directly attributed to these vascular problems. So it's a really important problem to solve, is identifying who's got these problems and treating it. That's why I founded the Neurovascular Modelling Group. Our aim is to develop imaging and analysis methods that detect neurovascular dysfunction non-invasively, that are sensitive early in AD progression before any structural markers that would appear on a normal brain scan. It's really important they can tease apart neural problems and vascular problems because they are likely to have different treatments. And although we're going to use really advanced laboratory equipment for doing our studies and understanding the conditions, we have to bear in mind that for any future clinical translation, all of this has to eventually work on standard equipment you could find in a hospital, like a standard MRI scanner. If we can achieve that, if we can have these methods, it would allow us to answer lots of really interesting questions. And just to give you a flavour of them, it would allow us to understand AD progression better. So how AD is Alzheimer's disease. How does vascular dysfunction fit in the progression of Alzheimer's disease? Why does it appear early? Is it causative? This condition I mentioned, CAA, can we spot it early enough that it can be treated? There's all these very exciting new immunotherapies that can clear out amyloid from the brain, but they have a real complication. A lot of people who take them get these microbleeds. Could we identify with a proper kind of diagnostic test who's got very strong and healthy blood vessels who are unlikely to get those complications and steer them towards the treatment appropriately? Clinical trials, can we provide readout measures? Can we give a simple number, essentially, to a clinician or a clinical scientist to say, here's how healthy someone's neurons are, here's how healthy their vessels are, and have those numbers have meaningful units that they can interpret? There's also really exciting new biomarkers, like blood biomarkers, that can detect these molecules without requiring expensive brain scans. But we want to know what the numbers mean that come out of them. So could we use these techniques to understand what a high or a low value, for example, of tau in the blood might mean, or amyloid? So that's what we're trying to do. That's the aim. The team is me and a postdoc named Pranay, who recently joined us from Cambridge University. He's a real expert both in physiology and in mathematical modelling. A PhD student, Josie Linnell, who's going to be starting with us in October. And then this is really a multidisciplinary aim that we have here, and it requires several teams working together. So from the Dementia Research Centre, we've got Nick Fox and John Schott's teams. The physics team, led by Martina, who can do magic with MRI scanners, are absolutely crucial here, and Rick Henson's team in Cambridge. And I want to just take this moment just to say a few quick thank yous. The road to getting funding in science is not smooth, and I undoubtedly would have given up sooner if it hadn't been for Cathy's gentle mentorship and telling me to keep going. Jason Warren has been a fantastic mentor in terms of the clinical side of things, helping me to understand not just what a scientist is interested in, but actually what a clinician would care about and what is useful, which has kept me really grounded. And Martina, who is an absolutely wonderful line manager, leader, mentor now, and has been instrumental in setting up these collaborations across our departments. So thank you for making all of this happen. So that's thank Medical Research Council as well. Thanks. Much appreciated. Much appreciated. I just want to emphasize, although I'm talking about Alzheimer's, everything here has wider application. So any measures we create are useful for a whole load of different conditions that affect both the neurons and the blood vessels. And there's quite a few of them. And all the tools we're making, we just make freely available in SPM so that any clinical researcher can use them. So that was the aim, the big ideal. Now the question is, how do we do it? And this is where we come back to what I started with, the mathematical modeling. And I'm going to allow myself to get a little bit more technical, but I'm going to try and bring everyone along. But there will be one or two equations, but I'm hoping you are going to enjoy them. Right. We're going to need 3 ingredients. We need multimodal imaging, we need generative models, we need Bayesian statistics. I'm going to explain what those 3 things are. Remember the aim. We want a non-invasive assay for neurovascular dysfunction. And as I mentioned, there's 3 components. We need to be able to measure those blood vessels, to what extent their dynamics are healthy, if they're moving well. But the trouble is that the blood vessels depend on both their own health and also the signals they're receiving from the neurons. So that means we also need an independent measure of the neurons in addition. And once we know those 2 components, we can infer the third part, the neurovascular coupling, and ask if it's working as it should be. So to do that, we're using 2 different kinds of imaging. The first one is functional MRI. What we're doing is we're showing pictures that look a bit like this, black and white checkerboards, that are designed to activate the back of the brain. And we're interested in the back of the brain because that's where you see the biggest effect of vascular disorders. Those images go through some mirrors and are projected into the MRI scanner. And then we take a measurement every few seconds. And what we can then do is build up a time series for every point in the brain that reflects the level of activation. We're then having the same volunteers undergo magnetoencephalography. So this MEG is an approach that looks like a large 1950s hairdryer, and it directly measures the activity of neurons. Each neuron or each pyramidal cell, more specifically, acts a bit like a bar magnet, and you can read the magnetic field that it gives off. So we end up with a time series for the fMRI and a time series for the neural recording, the electromagnetism. The populations, the people that we're going to have volunteer, we're working with 2 populations at the moment, but there's others that will be coming on board. First, we're working with Nick Fox's team and Natalie Ryan's team at the Dementia Research Centre, people who have got this condition called cerebral amyloid angiopathy. And we're also working with collaborators in Cambridge University who have this massive study, 700 volunteers, who have undergone both kinds of imaging across the full age range from 18 to 88. And these 2 data sets are going to allow us to understand what's normal for aging and what is abnormal with vascular conditions. As I mentioned, we get these 2 measurements. The vessels we measure using MRI, the neurons we measure using MEG. This is at a research stage. When we eventually come to clinical translation, we'll only have the MRI. But at this point, this helps us to understand that MRI signal. The question now is, how do we put all this together? Because essentially, we've just got wavy lines. So how do we go from wavy lines to a statement like, there is a 90% chance this person has a problem with their blood vessels, and only a 10% chance they have a problem with their neurons? To get an inference like that from the lines, we have to analyze the data. And that requires some models, and it requires some statistics. The first thing we could try is we could just describe the data. So we could measure the signals that we could get. We could measure the height of the signal. We could measure the slope, and people often measure, this is the MRI signal, people measure this slope, its angle. Or we could take lots of signals from different points of the head and we can compute their correlation. That is very common, and it's very common in clinical imaging. And you could say that people with a disorder versus without have a different slope or a different amplitude. I would suggest that that is a very limited, that's a first step, but it's limited in terms of how much we can interpret. Certainly, if I went to a clinician and said, "Doctor, this patient has a 43-degree slope in their MRI signal," they're going to say to me, "So what?" And I think that's part of the reason why functional MRI hasn't had such a big take-up in clinical imaging, because the research studies typically do not go beyond this descriptive level of trying to understand the signals. So if we're going to go beyond describing data and move to explaining the data, that's where we have to have models. A model is just a mathematical device that says how we believe the signals that we are looking at were generated. So this is the form of the models that we typically use in my group and in the methods group. Here we have a stimulus, so that's the image, the picture that was shown in the MRI scanner, which drives the neurons, which drives the blood vessels, and drives changes in variables or quantities that are really interesting, but that we cannot directly measure. Things like the cerebral blood flow, so how quickly is the blood flowing, or the cerebral blood volume, how much blood is there. Those are things that would be interesting to a clinician, but you can't directly measure them. They're locked inside the head. What the model can do, though, is it can describe how those latent or hidden variables give rise to the measurements we make. And, because we're also collecting direct measures of neurons, this can also say how the EEG or MEG signal is being generated. The power of having a device like this is that I could try turning up or down the stiffness of the blood vessel, and I could predict how that changes the data we'd expect to measure. And what's the point of that? Well, the point is we can then run it in reverse. So we can start the model from the data, work it backwards, and say in this specific patient, their cerebral blood flow was 90% of what we would expect of a typical individual. And that then becomes a useful inference that has meaningful units and could be described to a clinician. I'm looking at the time and thinking, how much can I spare you mathematically? Let me just tell you a tiny bit, because at the beginning I said, this is all about maths. I just want to put a little bit in just to show, because I think there's some people in the room who do model data, and it might be interesting and useful for you. The key mathematical machine that I use in my daily work and have done all the way through is a framework known as state space modeling. This came from engineering. NASA, working with a man named Callaghan, some of you have heard of the Callaghan filter, invented this formalism in order to get spaceships to the moon. And the idea is that it distinguishes two kinds of things. So it breaks down the model into two parts. Two equations. The first equation is all about those variables I mentioned earlier that are interesting but we cannot directly measure. Things like blood flow, blood volume. It says how those values change over time. Crucially, it separates that from the stuff that we do measure but that is not interesting, like the noisy, wavy lines we get from our imaging machines. And by splitting these two components, you now have a formal expression, a description, a story about how the interesting stuff in the head gives rise to measurements that you can then run backwards and go from measurements to interesting stuff. Mathematically then, once you've got this, it allows you to leverage and utilize a very large literature from the engineering community that's also useful to us as neuroimaging people and neuroscientists. And to give one example, you can go from the model to the kind of descriptive statistics that people use in neuroimaging. Things like correlations, aka functional connectivity. Things like coherence, which is used in electromagnetic measurements. So we have a complete model that goes all the way, say, from the stiffness of a blood vessel, which I can turn up and down, to the measurements and the descriptive statistics that people tend to report in papers. And that means we can link them together and infer the underlying mechanisms that gave rise to our data. Just in the interest of time, I'm going to skip this, but the bottom line is that there are statistics that are actually surprisingly simple, which enable us to say whether one model is better than another. And that's absolutely crucial to the work we do. Sometimes these statistics are hard to get at. They are in the clouds, and we have to use some interesting approximations to try and get hold of them. Those approximations don't come from neuroscience. They come from other fields of study. And one of the really important contributions that Carl has made to the field is bringing in techniques that have come from machine learning and statistical physics, And that's happened both at the Phil, Carl's team, and also at the Gatsby computational unit at UCL, bringing in these methods that started with Richard Feynman in statistical physics, combining those with the kind of state space models I mentioned, and giving us the ability to come up with these numbers that just tell you at the end of the day whether one model is better than another. What I've been doing for the last quite a few years, before I started the neurovascular modelling group, was applying this framework in lots of different settings, because it's very, very flexible and can be used for different things. I'm not going to go into any detail of those applications, but these have been collaborations that I've really enjoyed and where I have grown as a scientist. I worked with people at the NIH in the States, Chris Baker and Ed Stilson, to apply these methods to look at visual receptive fields and gain probabilistic inferences about how the visual cortex of the brain is laid out. I worked with Francis Jin, who's based in Hong Kong, Roman Kotov, based in New York, to apply these same methods to the development of neuropsychiatric disorders and symptoms, explaining how disease processes give rise to symptoms. They're particularly interested in conditions that oscillate, where you have symptoms that come and go over time. And we were able to use this framework to try to understand what is the typical orbit or trajectory of symptoms that then allowed us to predict when the next onset of symptoms would occur. A really interesting project, well outside of anything I would have previously thought about. Johan Medrano, my former postdoc, now in Boston, used these methods to do some really interesting work looking at naturalistic imaging. So imaging where we're trying to understand the brain in its natural state, in this case exploring a virtual reality town, an experiment led by Eleanor Maguire's group. And Johan was able to use these methods to infer remember I said it's all about variables that are interesting that we cannot directly see. Well, the thing he was interested in was the connection strengths between certain neural populations. And he was able to infer the time-varying strength of these connections from data that was recorded while people experienced the virtual reality town that they were walking around in. Really interesting experiment. Back in my group, and back now closer to the experimental question of interest, my former postdoc Amir Jafarian was using the same methods to ask, could we integrate neural signals that we measure with MEG and the fMRI signals we measure with fMRI and put them all into one big model and then infer the neurovascular coupling. And his work showed that that was possible. A lot of these different projects have given me an understanding of what's useful, what's not, and how to explain it. And explaining this stuff isn't always easy because it relies on understanding about neuroscience, dynamics, statistics. I mean, it's frankly terrifying. So I've spent quite a lot of my time trying to write tutorial papers that simplify these ideas and try and explain it where possible in as close to plain English as I could achieve. And I think these papers have, well, at least been useful for a few people. So what we're doing now, and I'll just finish with this. The work we're doing now is to take remember I said at the very beginning, this is about making simple stories of how the brain works. So the simple story we have of how the vasculature works is shown in this image. What we're doing now is making these models fit for purpose for understanding disease and disordered neurovascular function. The idea is to have parameters or numbers that you can get out these models and present to a clinician. And Pranay is doing some really exciting work upgrading different parts of this model and then using very realistic models of brain vasculature that are derived from mice that are realistic to validate the inferences that we make. So those are the three ingredients. So in summary, I started by saying some thank yous and to so many different people, to my parents, to my family, to my friends, mentors, and colleagues. I talked about the methods group, and bottom line I hope to get across is that SPM and the methods group are in a very healthy state, and there's a lot of really exciting stuff going on, especially this year for our 30th anniversary. And we're just getting started in the neurovascular modelling group, and hopefully what we're going to come up with will be useful for the community. In the last minute, if it's not tremendously grandiose, and it might be, I'm going to give some unsolicited advice for junior colleagues who are in the room, based on things I've picked up along the way. Number one, change fields or research topics once. That means that you can bring something new that others haven't brought to a field, but doesn't mean that you're changing too often. Maybe twice, once or twice. Remember that experiments are only as cool as the hypotheses they test. This is something that comes up in the methods clinic all the time. Just because an experimental paradigm is cool, interesting, shiny, or indeed a model or a mathematical technique is shiny, exciting, cool, it doesn't mean it's in the slightest bit interesting for understanding the brain. Only do stuff where there's an interesting hypothesis to be tested. Take every opportunity to practice public speaking. Being able to communicate the work you do is as important as doing the work. Otherwise, it will never get used. Get to know your colleagues. They're amazing, at least they are in Queen Square, and have fun. And I'll leave it there. Thank you very much.
Wow. What an amazing lecture. I mean, it's really a credit to you, Pete. And you've shown us yet again how you can take really complex concepts and break them down and explain them to us in an accessible and an engaging fashion. And it's a real credit to you. You've also shown us, as we've come to kind of expect now, that you can pop in quite a lot of fun and humour into your lectures. And I think that's really fantastic. Comic timing is one of Pete's many varying abilities. And some of you may know, some of you may not know, that he's also been quite brave about this. He's taken to the stage on comedy nights and put himself out there, which I think is a really, again, a credit to him. But what I also think is kind of that's part of is his remarkable appetite for growth. And not just in an intellectual context, and of course you can see that with all of the different things he's done and the bravery of changing topics and things like that, but also in his personal life. You know, he puts himself out there. He and his friends take opportunities to pick a different activity that's going to put them outside of their comfort zone. And I just think, like, what a remarkable way to live your life, you know, always seeking growth, new opportunities, stretching yourself. It's a credit to you. Doesn't always go well. We know this. You can ask him about his recent camping trip, although I think you probably don't need to ask him because you can probably guess that wasn't really suited to our Pete. But fair play, you did it anyway. So I think that's the credit to you. Pete's appetite for growth is one of his many wonderful characteristics. I find him to be such a diplomatic, warm, open sort of person. And I think what we have with that is a real clarity of thought and a depth of knowledge. And putting those together you think might be enough, but no, Pete goes further. He's incredibly generous with sharing that knowledge with all of us to the benefit of all of us. And I think he epitomizes what it is to be at the Phil, where there's this collection of knowledge and an effort to always be more than the sum of our parts by working together in a very collaborative way. And I think that that great sense of community is actually inspired by the way that Pete approaches things with openness, friendliness, generosity. So you're a huge part of that culture. So we're all very lucky that the sensible software engineering consultancy route was abandoned for the very exciting world of neuroscience. And we're very grateful to Cathy Price, who you may not know, but Pete reached out to her about maybe doing an MSc project, and she got back to him very quickly and seized what turned out to be a wonderful opportunity for you, but also for the rest of us. And Pete has had a lot of really formative foundational figures from the Phil who have been a part of his training over the years. Cathy, also Carl, and of course Eleanor Maguire. And you know, Pete, I know she would be incredibly proud of you if she could have been here today. And she would have really enjoyed seeing your lecture and seeing what you have achieved, you know. And maybe one drink, Pete, could have even prompted her to enjoy a little celebratory tipple with you. Who knows? But what she'd also say is, onwards and upwards, don't rest on your laurels, right? And she'd be looking to see what achievements would come next. And I'm sure there will be many, and that they will be impressively impactful, just as everything that you've achieved so far has been. So Pete got training from foundational people, but now he himself is part of the foundations of the future at the Phil, which I think is just really something wonderful. He's moved phenomenally in his transition from student to master. And he shows so much leadership, for example, around our project presentations, which he chairs. These are a key part of our scientific governance. And every week he launches them with, "Happy Friday, everybody." And it sets the tone for a lovely, fun way to end the week with some interesting science that's to come. It's not just that, it's chairing the methods group, engaging with the methods clinics, with the SPM Shore courses that are run. And these are a key part of our international training offering at the Phil. And then it is also the development of SPM, which has been a key offering both from the past and for the future that Pete has been chairing so well over these past years. And again, like, it's not each of those kind of diverse enabling activities in and of themselves would be remarkable achievements, but you're doing all of that with a big smile on your face, while you're also then developing your own research direction by establishing this neurovascular modelling group, which again is a real credit to you. And then reaching out to all of our clinical colleagues around Queen Square, fostering these new collaborations and really making the utmost of what it is to be in this amazing environment. So I'm going to end my vote of thanks with a quote from Carl, which I think summarises how impactful Pete has been and how happy we are to have him in our ecosystem. Carl says, "Peter is a godsend. Anybody who has worked with Peter will know exactly what I mean. He is a key personality, an intellectual pillar upon which the Phil rests." So from all of us, Pete, thank you very much.
So we're on to Peter Kok now, who's our second professor from the Phil. I'm sure Peter's going to go through his story a little bit as well, but it's just the geography's pretty impressive. So he started off also in computing, I think, at the University of Groningen, then moving on to do some other degrees there in Cognitive Neuroscience, and then moving on to Nijmegen. Before moving on to the Donders, then Princeton, some of you may have heard of that, and then Yale, before coming to the Piece de la Resistance, UCL, which he hasn't left since, much like Peter before him, coming to the Wellcome, and then getting a very prestigious Sir Henry Dell Fellowship to enable him to continue his work. He's not published a huge amount of papers, and this is a slightly controversial view. I think you can tell a very serious scientist by how few publications they have, actually. So he's got 46 publications and over 7,000 citations, and I'll leave you to do the maths on that. So he doesn't speak often, but when he does, people listen and cite it. More recently, he's had several awards. Last year, the Cognitive Neuroscience Society Young Investigator Award, despite obviously having a lot of young people around him as well. And more recently this year, the Early Career Impact Award from the Federation of Associations in Behavioural and Brain Sciences. So Peter's going to talk to us about priors and visual perception. So we're moving a bit from methods more into the application of imaging neuroscience. Thank you, Peter.
Hey, thanks everyone for coming. I'm gonna jump straight into it. So we're gonna talk about visual perception now, using a lot of the foundations that Peter just set out in terms of the methodologies that are developed of inferring things about the human brain. Now, looking at how we can use those techniques to look at visual perception. So how did it all start? Well, it started, of course, with a general sense of wonder about the world. But very quickly, it became a lot more sophisticated than that. And then started wondering, I don't remember when, but at some point started wondering how this kind of rich experience that we have of the world and experiencing all these different thoughts we have and perceptions, how those can arise from basically this lump of meat in our heads, the brain. How does this work? That was the kind of the big question for me during my early studies that motivated the questions I had, like how does this all work in this brain that we all have in our heads? So this fascination with our rich consciousness also developed into an interest in literature, particularly the types of literature that try to describe this very rich experience that we all have. One example quote from a Dutch book that has been translated into English as well. I won't read it out, but it's particularly apt for the kind of topics that we'll talk about today. So this type of literature describes the richness of the mind, which I find fascinating, but I quickly realized I didn't have the talent or the intelligence to contribute in this way to understanding the mind. So I decided to change tack to doing a bit of philosophy after doing a bachelor's in psychology, which actually, as Alex said, followed a year of computer science, which I then abandoned, but much earlier than Peter Zeidman, because that turned out not to be my forte. I switched to psychology and then a bit of philosophy of mind to try to see if we could make sense of how the brain and the mind are supposed to work in a physical world. And one thing I did as a little thesis during that course was to look at how causality is supposed to work for a mind in a physical world. And the basic idea here is that we presume that mental states cause other mental states. When you feel hungry, you have thoughts of delicious food you might want to eat. But in a physical world, those mental states must in some way depend on physical states, brain states, presumably. That's true for both of those mental states, the cause and the consequence. They both rest on a physical state. So where is the causality in this? Where does the causality go? Do mental states cause mental states, or do brain states cause brain states? That's a tricky question to answer, especially if you don't want to reduce mental states to brain states, because then you presumably have to give priority to the physical world if we want to take the physical world seriously. But that then means that mental states just kind of sit on top of that without any causality, any causal impact, like kind of the steam from a steam engine, that kind of epiphenomenon, which is not very satisfying, of course. And what the argument that Jae-Gon Kim makes in his book is that all of this is solved if we accept an identity relationship between mental states and brain states. If we say mental states are brain states, then the problem goes away. The causality goes from mental states to mental states and from brain states to brain states because they're the same thing. So after considering philosophy for a little while, I also realized this is also not the best way for me to contribute in terms of where my skills lie. I did find this very interesting, but it's not the way I thought I could contribute to learning more about how our consciousness works. I did think this does mean, I thought, this little essay exercise, that if mental states are brain states, if we take that seriously, then studying the brain might lead us to might help us understand a bit better how the mind works. So that's what I then pursued further in a research master at the University of Groningen, the Brain and Cognition and Neuroscience research master. This was a new research master's at the university at the time, I think maybe the second year or something. And we were a small group, but this turned out to be really great because we had a lot of interaction with the professors. We had a lot of interactions with each other. We learned a lot from each other. And this turned out to be a really great program for me to develop my interests further. And the great thing about this program, one of the great things was that it had 2 years, and in each of which year you did a big research project. For my first year, I reached out to Professor Ritske de Jong. And I basically wrote to him something like, I had read a bit about neuroscience and consciousness, and I wrote to him that I would like to do these experiments. I had some ideas of what kind of experiments. And with great surprise, in hindsight, he was very happy for me to do whatever I wanted to do and support me in that. And not only that, he also agreed to buy a stereoscope, which is this device with mirrors, which allows you to present two different images to each of the two eyes. And for instance, we know what I had read from a bit of reading I had done is that if you do that, if you present two different images to the two different eyes, in this case horizontal and vertical lines, you don't see both at the same time. You can only see one. And what happens is you see one for a while, and then you transition to seeing the other one, and then you go back to the original one, and you go back and forth. So the input to the eyes and the brain is constant, but your conscious experience fluctuates over time. I thought this was fascinating, and I wanted to investigate this further. And I did so during this first project with a lot of help also from Paolo Toffanin on the top right, Rizke's PhD student. And we had people do these kinds of experiments while they wore an EEG cap. And we did like four little mini experiments, one with these kind of oriented lines, one with faces and houses, one with depth perception, all kinds of little experiments. And then we tracked how the brain signals fluctuated along with people's conscious perception of these stimuli. I had a lot of fun. I learned a lot about setting up an experiment, analyzing some neuroimaging data, interacting with these great scientists and programming a bit. So I learned a lot, but we didn't necessarily learn a lot from this about consciousness or how the brain works, for reasons that Peter Zeidman already explained in his lecture in point 2 of his advice to junior scientists. There's no hypothesis testing here, really. This was just playing around, seeing what happens in the brain when people see different things. So that brings us to year 2, which was about testing hypotheses, a very different kind of project, together with Jacob Jolley, which his nickname, his English pronunciation of his name, Jacob Jolly. This was a great project which was completely different. Jacob had a project that he had thought out based on work he had done before. And this involved doing transcranial magnetic stimulation, which is a technique to modulate brain activity from the outside using magnetic coils. In this image, they're on the top of the head, but in our case, we stimulated the back of the head where the visual cortex is. And when you do that, people see flashes of light. And what Jacob had found in his previous work is that if you present people with an image, in this case, vertical lines against a backdrop, and then you stimulate their visual cortex a little bit later, people will often see the image twice. They will see the vertical lines appear again at the time you stimulate their visual cortex. So in that case, they won't just see a flash, they will actually see the image they had seen before again. And then the question that he had and that we addressed together was, how does this work? Is this a kind of passive reawakening of this previous stimulus that had just been presented? Or is this because you expect to see vertical lines, because you have been seeing vertical lines throughout the experiment? If the latter, then results should be different if you're not as sure about what you will see next. So what my experiment involved is not having just one possible image, but in different conditions instead of 2, 4, or 8 images. If this is about expectation, you should have less of these percepts in the 8 set condition, because there are 8 possibilities, you're not sure what to expect. And that's indeed what we found. As the set size of the images increased, the number of these kind of false percepts, if you want to call them that, decreased, showing that it's not just a passive reawakening of a previous stimulus. So not an experiment that answers all the questions we have about consciousness, but it is an experiment that tests a hypothesis about how something about perception works in the brain. And I think, so here you might say, the field learned a little bit from this. So this, I think, emphasizes the importance of, as Peter also did, testing hypotheses. So then, after this master's program, I moved on to do a PhD with Floris de Lange at the Donders Institute in Nijmegen, which was a great opportunity. And something I want to emphasize is that all these places I've been, including here, is not just about working with one person, one supervisor, but it's always a whole community of people. In this case, at the Donders, loads of people in our own research group, collaborators from different research groups, people, cognitive neuroscientists, but also people from methods groups. And this whole community makes a successful neuroimaging program, and that's been a great experience. So in this PhD with Floris, I worked on the role of expectations in visual perception. So I want to explain what I mean by that with some examples. So if you look at these two blurry shapes here, they probably don't really mean much of anything to you if you're just looking at them in isolation. If I put them in their proper context of this blurry street scene, then you'll see that you're looking at a pedestrian in the foreground on the right and a car in the background. Yeah, a car. And if you then take these blurry shapes out of their context again and you rotate them so they have the same orientation, it turns out these two shapes are actually identical. There is nothing about these shapes themselves that makes them a pedestrian and a car. It's about the context they're in and their orientation in that context. So it's really our knowledge of street scenes that makes us see these shapes as we do. This is a sculpture on the street in Toronto, a hollow carving, as you can see. If we move across it, if we walk in front of the sculpture, at some point we'll face it head on, and you might have this perception of the face popping out of the sculpture and looking like a convex rather than a concave face. This is known as the hollow mask illusion. It works with lots of different stimuli. If you're looking at the inside of a mask, you'll have this experience as well. And this is presumably to do with the fact that we expect faces to be convex, not concave. This is what we've been exposed to our whole lives. And this kind of prior knowledge of what faces are like overrides the fact that we actually know we're looking at a hollow mask and makes us see it as coming out of the stone. Final example. If you haven't seen this image before, you probably assume you're just looking at a brick wall here with some mortar. If I highlight this image here in the center and blur out the rest, you see you're looking at a cigar here. And now if I move back to the original image, you see that what we've been looking at is a brick wall with a cigar sticking out of it. And now that you've seen this, next time you'll see this image, you'll see the cigar straight away from the start. So your knowledge about what you're looking at changes your perception of what this thing is. Okay, so those are some examples of when prior knowledge and expectations change the way we see the world. In our studies, unfortunately, we don't use these kind of fun examples, but we use gratings a lot of the time. So why do we use gratings? Well, of course, because they're very exciting, naturalistic stimuli, as we can see here. They instantly draw our attention. So clearly they're the best kind of naturalistic stimulus to use in an experiment. But the real reason for using these gratings is slightly less fun maybe, but it turns out that these kind of oriented lines are one of the kind of basic features that the brain deals with when processing visual signals. So from early studies in the 60s by Hubel and Wiesel, we've know that neurons in the visual cortex, they are tuned for particularly oriented lines, and they fire when lines are presented with those orientations and not others. Coming at it from a different angle, from more a computer science angle, if you train a neural network to process images and you ask it to be as sparse as possible, so to represent them with as little activity basically as possible, it turns out that these models often develop tuning for oriented lines or gratings. So coming at it from different angles, it seems really that these oriented lines are a basic tuning function of the visual processing system. So what we figured in our work was, if our prior knowledge and expectations change even the processing of these kinds of basic units, then that must mean that they're really fundamental to the way the brain processes visual signals. So my first experiment that I did during my PhD involved trying to look at expectation and attention and see whether they might be different things or the same. And the way we did that is a really simple design, where we told people that, for instance, in this next series of trials, images are mostly going to appear on the right side of the screen. Then we gave them another instruction that said, you only have to pay attention to the left side of the screen. If the stimulus appears on the right, you can ignore it. If it appears on the left, you have to do a task on it. And then, for instance, in a given trial, in this case, the stimulus appeared on the left, which meant people had to do a task, but it appeared on the unexpected side. Another example trial here, expected on the left, pay attention to the right. In this case, the stimulus appeared again on the left. Now it was expected, but ignored. We had people do this task while we scanned their brains in an MR scanner, and then looked at the activity in the early visual cortex, using all the kinds of techniques that Peter just described, but in the kind of more amplitude slope style, at least initially. And what we found is that for stimuli that could be ignored, that appeared in the ignored side of the screen, they evoked less activity when they were expected and more when they were unexpected or surprising. But for stimuli that people had to attend and do a task on, it was the other way around. They evoked more activity when they appeared on the expected side. So it seemed that expecting something has very different consequences for visual processing depending on whether that stimulus is relevant to the task you're doing or not. So we could make some sense of that kind of informally, but we wanted to try to understand it a bit better. And to try to understand it a bit better, we turned to important theory in visual processing, which is predictive coding. First applied to thinking about the visual cortex in the late 90s, with this proposal that what's happening is predictions are being compared to inputs, and then the mismatches generate prediction errors. And then later by Carl Friston, a much more sophisticated version of this where this gave a lot more pointers about how the brain might be doing this and incorporating important things such as precision, how do you weigh the reliability of those signals, etc. So I'm not going to go into the details of the theory, but what we did is we did some very simple simulations to show that this theory actually explains that kind of crossover that I just described in the activity very well. So this theory can explain this phenomenon I just described in a formal way very well. We also looked at the connectivity between the visual regions as a consequence of modulations in predictions, etc., using a technique again described previously by Peter, known as dynamic causal modeling, also developed by Carl and with great contributions by Peter in recent years especially. So this technique allowed us to try to gain a bit more insight into what's going on in the visual system during predictions. And then after this study had finally been published, after going along number of different journals, I got this email from Carl 2 days before Christmas saying that he had read my paper that had just come out and that he liked it very much. And you can imagine, as a PhD student, having published your first study and then getting an email like this from someone who inspired a lot of the work was amazing. So that had a big impact on me then. So we did lots of other work during my PhD to show, for instance, that expecting something can evoke a template of that expected image in the visual cortex, even if nothing is presented to the eyes. And those templates can be evoked prior to stimulus onset, so before anything appears, so that you can kind of intercept the upcoming stimulus and process it better. When your predictions are right, when they're valid, this leads to more efficient processing. So expected stimuli then evoke less activity overall, but there's more information in the signal about what the stimulus was. And when expectations are wrong, and the stimuli that you are presented with are noisy, so hard to interpret, then expectations can actually bias what you see. They can change what you see, and they can also change how those stimuli are represented in the visual cortex. So those are some things we did, but we didn't only study gratings, fortunately. We also studied this visual illusion known as the Canisa illusion. Here, this looks like a white triangle superimposed on 3 black circles, but there is no real triangle there. This is presumably the cause of the brain dealing with occlusion all the time. So if I hold up this water bottle and include it with part of it in my hand, you're very well able to know how the bottle continues behind my hand. Occlusion is a very common thing for the visual system to deal with. So assuming that this kind of stimulus is not an arbitrary arrangement of Pac-Man-like shapes, but is caused by 3 circles with a triangle on top of it is a very, very parsimonious explanation of the inputs. And just to convince you that this is an important illusion, it also it has been studied in cats. So it turns out cats, they, as we all know, probably they really like sitting in boxes. And that extends to not actual boxes, but also squares taped on the floor. They really like sitting in those. And what people did in this study is to tape Pac-Man shapes to the floor, either in an arrangement that causes an illusory square on the right, or with the Pac-Man rotated so that there is no illusory shape. And it turns out that, yes, cats prefer sitting in illusory squares over sitting in other arrangements. So I hope that's enough to convince you this is a really important illusion. So we studied this in the human visual system. Basically, our question is this triangle that's not actually there, does it still evoke activity in the early visual system? So again, we put people in the MRI scanner and compared these illusory triangle arrangements with rotated arrangements where there's no illusion, and looked at the early visual cortex. And we found that in two different conditions, that indeed at the location where the illusory triangle is perceived, in the dashed white triangles here, there's increased activity when you have this illusory percept compared to when you don't. So in this part of the early visual cortex where there's no actual input to the eyes, there's still an increase of activity when you think there's a triangle there. So that's some of the work that we did in my PhD, and then I stayed on a bit longer after finishing to do one more project that we really had been wanting to do since I started at the Donders. And it's slightly more technical, so I'll explain that a bit now. So basically, in all that we've done so far, we've assumed that the cortex is a two-dimensional sheet. So kind of flat like a pancake. But actually, it turns out it's better to think of the cortex as a stack of pancakes. So the cortex, actually, when you look at it, is not just a two-dimensional structure, but it has depth to it. And that depth is important because at different depths of the cortex, there are different types of neurons, there's different vasculature, and importantly for us, a different connectivity with the rest of the brain. So signals coming in from the eyes, if we're talking about the visual cortex, arrive predominantly in the middle layers, and feedback arrives at the very superficial layers and the deep layers avoiding the middle layers. So again, we can go back to the stack of pancakes. If you imagine that you're pouring syrup over the pancakes, a lot of syrup hits the top of the stack, and syrup pools at the bottom, affecting the bottom of the pack of pancakes, the deep layers, but the middle pancakes stay relatively dry. So this is important for us because it can tell us, if we know which layer signals are in, this can tell us something about where they likely came from. The tricky thing, though, is that the cortex is about 2 or 3 millimeters thick, and that means it's very difficult to tease apart these different layers when studying the human brain. But what we did is, using a 7 Tesla MR scanner, boost the spatial resolution of these functional measurements so that we have about 3 voxels over the depth of the cortex. So that allows us to roughly separate the neural signal into superficial, middle, and deep layers, which is enough, at least initially, for our interest in trying to separate these different compartments. And our first effort at doing this, we turned again to this Canisa illusion because it had been a very reliable a paradigm that worked very well. And we were very curious to know if we can tell where this illusory triangle activity, where that comes from. So what we did is again compare arrangements where there was an illusory triangle compared to where there was none, and then focusing on the part of the visual cortex that responded to this illusory triangle location, and found that specifically in the deep layers of the early visual cortex, there was increased activity when the illusion was there. So if we go back to the other way of visualizing this, you can see that this illusory triangle activity is really there in the deep layers of the visual cortex and then goes away as you go through to the middle and the superficial layers. So this suggests that this illusory activity in the visual cortex is a result of feedback from higher order areas. So this result itself was exciting to us, but also the potential that this gave for answering new questions in cognitive neuroscience. Being at the Donders was a great time for doing all these different studies, but it was also a great time for other reasons. I met my favorite co-author at the Donders, who's still here today and supporting me through all this, and who's still also, despite having left academia in 2016, his citation record is still going very strong. So it was an amazing time to be at the Donders, and ended with defending my thesis in front of an audience, kind of in this kind of setting, supported by my colleagues and friends, and being examined by Carl. See him here on the photo on the bottom right, leftmost. That's Carl again examining my thesis. It was a great honor to have him examine my thesis at the end of this work. Okay, so then after spending all this time at the Donders, I shifted slightly in terms of the questions I was trying to address. And the question that I focused on next is not just how does this feedback affect the visual cortex, but where does it come from? So if we go back to the stack of pancakes metaphor, where does the syrup come from? And of course, anyone who knows anything about syrup and pancakes knows the answer is Canada, of course. So I next then went to work with Canadian cognitive neuroscientist Nick Turk-Brown at Princeton University and later Yale when the lab moved. And as at the Donders, this was a time where there was a great community that I benefited from working with and spending time with and learning from. And this was a great experience to enrich my scientific life. And what we looked at with Nick, as I said, is where do these predictions in the visual system come from? So this is especially a relevant question for the kinds of predictions that the visual system itself doesn't have access to. So let's say that you hear bark and you expect to see a dog. The visual system doesn't have access to this bark. It can't process the bark itself. So that expectation must come from somewhere else. So what might that source be? In our thinking was that this potential source must get information from the different sensory modalities to be able to combine them, and also send feedback back to those sensory cortices to send predictions. And it must be able to learn regularities between different signals, such as the bark and the dog, fairly quickly in order to be effective. So that led us to think that there might be an important role for the hippocampus in this process, which is, of course, generally thought of as a memory system involved in episodic memory. But we thought it might also play a role in perception, especially when it comes to guiding perception using these statistical regularities. So specifically what we thought could happen is that when you present a cue that predicts something, in this case a sound, the hippocampus might retrieve the associated objects, in this case a grating, and send a prediction to the visual system about what is likely to happen next. And we tested this in a couple of fMRI experiments, and we found some very exciting to us evidence that this may be, that supported this hypothesis at least. So the signals in the back of the head and the visual cortex were really dominated by what was presented to the eyes, but the hippocampus specifically seemed to represent predicted visual objects. So not what was presented to the eyes, but what was predicted by what just happened before. So some really interesting evidence that got us very excited about this topic. Okay, then the move to UCL happened in 2018. This was a very exciting move for me because this was where I started to develop my own independent research group. And the whole department welcomed me and supported me greatly, which I very much appreciate. And I just want to mention a few people in particular. Cathy, who recruited me and spent a lot of time and effort mentoring me as well through this new stage in my scientific career. And Martina, the current head of the department and also a close collaborator on all these fMRI imaging methods. Steve, who I also asked lots of questions both before and after joining UCL, since he had a slightly similar position and similar grants he applied for a few years before me. So I pestered him with all these questions about how things work all the time, and he was always very patient in answering them. And Gareth, who's kind of the equivalent of Martina, but on the time-resolved front in all things MEG and EEG, which has been a great help as well. And Carl, of course, who's been shown up to this talk throughout. So this just shows you what the kind of perfect place for my research program the Phil really is, with these amazing methods people, but also the theoretical background that had underbuilt a lot of my work beforehand. And then Ellen Maguire, one of the world's foremost experts on the hippocampus, who I'm not sure if she ever really bought into my idea about how the hippocampus might be involved in perception, but that didn't stop her from always supporting me, both personally and professionally, and giving me feedback on all my grant applications, etc. And that was amazing to be able to work with her. And as Pete said, she's greatly missed by all of us. And one of my main collaborators from the start at the Phil has been Claire Press, at the bottom left, on all things expectation, especially where actions are involved. So this was really an amazing environment for me to land into and to start to develop my own kind of independent group. And the first pillar of that, and that's not even mentioning yet all the amazing support teams that we have supporting us with all the imaging at the Phil, the IT and the admin teams. It's really an amazing place to do your research. The first pillar of the research that we started in 2018 was Fraser Aitken, who in about a year did 3 neuroimaging studies using 3 different neuroimaging modalities, which I still think is amazing. He did a 70 fMRI experiment actually in Oxford, because it was just before the scanner here came online, showing that these templates of expected stimuli specifically show up in the deep layers of the visual cortex, so the same layers in which we saw these illusory triangles. He used MEG to show that expectations modulate sensory processing really early on at around 150 milliseconds. And he used 3D fMRI to study how expectations are learned in the hippocampus, showing that early on during learning, the hippocampus mostly seems to represent unexpected events or prediction errors, and then later, when learning is done, switches to representing predictions instead. So this is to show that this was an amazing effort by Fraser, doing all these different things and being super productive. We had then finished these studies, used these results in grant applications, and we're lucky enough to be able to award funding to start up a new round of neuroimaging work. And then, of course, the pandemic hit. And there was no neuroimaging work to be done for quite a while, which was, of course, a very frustrating time when you just think you're going to really get going. I think in February my grant started, and in March the first lockdown started. But there were also some great things that happened during that period. One of the highlights was a collaboration with Nadine Dijkstra and Steve Fleming. Nadine had joined Steve as a postdoc and was investigating how mental imagery affects perception. She had planned to do a lot of neuroimaging, but switched to doing this online, where participants sit at home and do experiments behind their computer just from the comfort of their home, which works during lockdown. And I found this very inspirational that Nadine was just able to keep going with her research in this way. And since then she has moved on to doing neuroimaging as well, and has gotten very exciting results, which I'm not going to talk about any further, because Nadine is now PI at the Phil as well. And I'm sure she'll be giving one of these inaugural lectures very soon herself. Then we started slowly getting back to neuroimaging, and the main driver for us really in our group was Joost Haarsma, who joined the lab in May of 2020, so during the first lockdown, and was really one of the first people who, together of course with the MR physics team, started collecting MRI data, fMRI data again at the center. And I want to tell you a bit about what Joost did. So we've seen so far that predictions selectively seem to activate the deep layers of the visual cortex, in the case of this Canisa illusion, this illusory triangle, but also in the case of predictions evoked by cues. And what Joost really wanted to ask is, can these predictions make us see things that aren't there? And the reason he was interested in that is background in studying psychosis from his PhD. And there are very prominent theories about how hallucinations work that suggest hallucinations are the result of excess kind of top-down feedback, internal signals into the visual system at a cost of visual processing information from the outside world, and that this results in an hallucination. Joost has also written about how you might be able to use layer-specific fMRI to investigate these kinds of phenomena. And a chapter that we wrote together just came out earlier this week in a new book about how you might be able to use these techniques to understand psychiatric disorders. So the work that Joost did was aimed at trying to look at false percepts. And the way he did that is give people, again, these kind of expectation cues. A tone predicts what kind of oriented lines you're going to see. But now the oriented lines are very difficult to see because they're presented very briefly and embedded in noise. And actually on half the trials, let's get a pointer here, on half the trials, there were no oriented lines at all. We just presented this kind of noisy image. But we still asked people to report what they saw. And what we then looked at is the signal in the early visual cortex, again in the specific layers. And as before, we found that these expectations, so the cue predicting what you're going to see next, led to representations specifically in the deep layers of the early visual cortex, consistent with the other studies. But what about these false percepts? So when people saw a noise patch and they didn't think they saw a grating, there was no real signal or any meaningful signal in the visual cortex. But then on trials where they were very sure that they had seen oriented lines, even though there weren't any, the orientation of those falsely perceived lines was reflected in the middle layers of the early visual cortex. And so different layers than the expectations. And also which oriented lines they kind of hallucinated was unrelated to the cues we had just presented. So we had expected that cueing them to, for instance, a left-tilted grating was likely to come up. They would be more likely to hallucinate left-tilted lines, but that wasn't the case. There was no relationship between the two. So that was surprising to us, and we still don't fully understand this. It makes some sense post hoc in a way of thinking about it like you don't want to walk around hallucinating your expectations all the time. So it makes sense to have some separation between these kind of top-down signals and bottom-up sensory signals. But what does this mean for these theories of hallucinations? One thing that's important to note here is that these expectations were very subtle. People were just trained on these expectations for a little bit in the lab. And actually, importantly here, we didn't tell people what the cues meant. We wanted to avoid that. We wanted to have people implicitly be exposed to these relationships and then see if that affects their perception. So that meant that these expectations were very subtle and people weren't even aware of them. So right now, one of the questions we're asking is, what if these predictions are stronger? Can they then evoke maybe a false percept of the expected thing? And this is something that a new research fellow in the lab, Gemma Donnelly, is investigating right now. This project is just starting up. We also have done more work on this on the hippocampus, since it's such a fascinating area to us. And one question that we've been studying using this layer-specific fMRI approach is, are these expectation signals that we see actually communicated to the visual cortex? So our hypothesis is that the hippocampus retrieves associated images and then sends them to the visual cortex to change processing there. But we don't have actual evidence for that. We have evidence that we see these signals in the hippocampus, but we don't have evidence that they're sent to the visual cortex. And one way of trying to get a bit more of a handle on the direction of communication between areas is this layer-specific imaging that I described earlier. And this is work that Oliver Warrington did during his PhD in the lab. So I'm not going to go into details of this circuit, but imagine that you hear a siren. What might happen then is that this sound is sent from the auditory cortex through the medial temporal lobe to the hippocampus, which then retrieves associated visual image of an ambulance, and which is then sent back to the cortex, to the medial temporal lobe, to the visual cortex. And the thing that's important here about this schematic is that these signals back from the hippocampus are sent through the deep layers of the medial temporal lobe and ultimately visual cortex, whereas signals going to the hippocampus go through the superficial layers of the medial temporal lobe. So if we're able to look at the layer-specific signals during predictions, we can infer which direction they're communicated in. So that's what Oliver did using the 7 Tesla MR scanner across the square at the Phil, using high-resolution imaging to look at the different subparts of the hippocampus and the different layers of the medial temporal lobe, and using a design where, again, a beep, an auditory cue, predicts what image you're going to see, in this case a shape. But now on 25% of the trials, the visual image is omitted. And it turns out that that omitted predicted shape can still be still represented in parts of the hippocampus and communicated to the deep layers of the cortex. So we have some evidence to suggest that indeed these prediction signals in the hippocampus are communicated back to the visual cortex to change processing there. And apart from this specific research question, I think it's very exciting to be able to look at the direction of communication between the hippocampus, one of the main memory systems of the brain, and the rest of the neocortex to investigate how these interactive processes work. Okay, so to conclude the science that we've done so far, prior knowledge strongly influences the way we perceive the world. The predictions are seen in the deep layers of the visual cortex, and depending on the exact type of prediction, these are provided by the hippocampus. And perception is really best thought of as the integration of prior knowledge and sensory signals. Either on its own is not really sufficient. You need to combine the two to have a successful percept. So why does this matter? For me, personally, this matters because as I mentioned from the start, I have this fascination of how does a rich visual experience arise from the brain. But if you want something a bit more applied than that, the understanding how this combination of prior knowledge and sensory signals works can really ultimately, I think, help us understand disorders of perception in clinical cases. So if you imagine there's not enough syrup on a stack of pancakes, not enough feedback, sensory signals may become impossible to digest and overwhelming, and you can't really make sense of the world. And there's been some hypotheses that this might be happening during autism spectrum disorder. And at the other side of the spectrum, again, this is in line with what Joost studied. If there are the prediction signals are too strong and they overwhelm the stack of pancakes, it just turns into a soggy mess. You might have percepts that don't make any sense at all. You might have hallucinations. And this is clearly also a disruption in perception that can be very detrimental and would be very helpful to understand better. And aside from just too little or too much internal feedback, there's also the potential of having the wrong kind of syrup, which might, in this case, be the wrong internal model of the world, which might also really affect the quality of your perception, that is your stack of pancakes. The present-day team of PhD students and postdocs and collaborators at UCL are working on lots of new questions here, which I'm very excited about. And I just want to very briefly highlight 3 of those questions that 3 PhD students in the lab have been working on. Dorothea has been working on how predictions are kept online in the brain. So you get a cue that predicts what you're going to see next, but you may need to wait a while before that outcome arrives. How does the brain keep that signal online? If neurons would have to keep firing the whole way through, that's very metabolically expensive. But what Dorothea has found is that it seems that these predictions are embedded in ongoing alpha oscillations in the occipital cortex, which is a much more efficient way of keeping a signal online than with constant neuronal firing. Peter has been working kind of on the flip side of prediction, post-diction. What post-diction is I think best explained with the example of the illusion that he's using to study this. First, a flash is presented on the screen, and at the same time, a beep is presented to the ears. Then just a beep, and then another flash and beep. And what people actually perceive is 3 flashes. they perceive an extra illusory flash in the middle, in between the start and the end point. And whether or not they perceive that illusory flash and where they perceive it is determined by the third beep flash pair. So what happens at time point 3 determines what you see at time point 2. So the present influences the past in a way. So that's kind of a mystery how that might work in the brain. And so far Peter has had one very exciting finding with this paradigm, which is that if we look at the different layers of the early visual cortex, it's the deep layers, which we're now very familiar with, where the activity resembles more seeing 3 flashes, so the actual perceptive people have, whereas the activity in the middle layers resembles more the presentation of 2 flashes, so actually the image on the retina. So these, again, some very suggestive evidence that these different layers of the visual cortex do different jobs. And finally, Zay is looking at predictions in a more naturalistic way. So far they've been very artificial, these paradigms I've been talking about with beeps and oriented lines, etc. In this case, people have a first-person view of walking along a scene, and then they hear a sound, for instance, the bark of a dog. They turn around, and then they might either see the expected object or a different object. And what we want to know is, do these much more naturalistic predictions work in the same way as those very artificial lab-based ones that we created, or are there important differences? And we think there probably will be some important differences. What those are is to be determined. So that's the present. We're also investing in the future, training the next generation at the Phil. So I think the future is in safe hands as well. So just to end on some take-home messages. Perception is like a stack of pancakes. Professors are not necessarily highly intelligent and good at everything, but they are highly curious, and that's what really is what drives a scientist and to keep at it. And it takes a village to raise a professor. So in addition to thanking all the colleagues that I've mentioned along the way of this talk, I just want to my family as well, for supporting my family and friends for supporting me throughout all this, a bunch of whom are here today, which I'm very thankful for. And I want to especially thank Alexa for all her support through this journey. And up to this point, professionally, Flynn has been more a hindrance than a help in my career, I would say. But I love him nonetheless. So thank you, everyone.
Before we get on talking about your talk and your work, it's so nice that you brought your village with you, and and it's been lovely to see you all, and everyone's been enjoying it, I can tell you. Yeah, so thank you very much. And I love seeing your baby photos as well, and also learning about your prior experiences that have shaped you and brought us here to Queen Square. So I'm very proud to have recruited Peter back in 2018, and I've had the great joy of witnessing him going from success to success, and now here we are celebrating his professorship. So I'll thank you again. What we've seen is that Peter's work and his talk here are really about how the human brain and also the cat brain can help us to learn to see the world. So that's obviously extremely clever and sophisticated. And most of us here will have had the past 2 years thinking how clever AI is. But AI just absorbs loads amount of information, and it doesn't have a sense of the context, and so it struggles and makes errors when the conditions change. So what we see here with the human brain is something far more clever, and Peter's talk reminds us just how special the brain is. What you've demonstrated in essence is how our brains minimize errors by dynamically adjusting our knowledge as we interact with the world, by building confidence in that knowledge, and paying attention to the unexpected signals that really matter. And you've demonstrated the most sophisticated use of all of our imaging equipment. You're the leader for the 70 fMRI, looking at the layer specific images. I love the pancake analogy that you have. That's really lovely. And really been a forerunner in the development of these in neuroscience. But what I wanted to also add was it's not just an academic exercise, but in the types of things that Peter is learning are really a beautiful illustration of who we are and what makes us human and what makes us much more than an AI machine, because none of this is in AI as yet. So on behalf of everybody here, I'd like to say thank you to Peter for updating our knowledge of human visual perception and for demonstrating why you so richly deserve to be a professor. So thank you, Peter.
Thanks so much, Cathy, and thanks to our candidates. So it just leaves me to wrap up. We're going to have a few photos with the new professors here, but for the rest of you, there's drinks. So please do stick around for a chat and some more discussions, perhaps, and perhaps just one last round of applause for both of the new professors, both Peters. Thank you.